for joining us for week two. I'm really excited for this portion. We'll be talking about the second part of honeybee biology, behavior, and physiology. So, to me, it's a lot of information, so it can be a little bit dense. But I think it's really exciting to see how maybe honeybees compare to like humans and other organisms. But before we begin, I want to talk about the Google Classroom a little bit because we had quite a few questions. So let me share the Google Classroom just to make sure we're on the same page. Okay, can you guys see the Google Classroom? I believe so. Okay. So if you're not part of the Google Classroom, please let me or Heather know. We can add you to the Google Classroom. I know some people had issues with certain email addresses to be on the Google Classroom. And so either you can change your email. It usually takes like an Outlook, Yahoo, or Gmail. But we've noticed that some business emails, they've had issues getting onto the Google Classroom. So it's just something finicky with Google Classroom that I'm learning about. So if you have any issues, just please let me or Heather know. To make up for that, if people aren't willing to make a new email address, I'll be sending out all the information after this lecture. So the reading materials, the quizzes, the study guides, and so on. Now, if you go under coursework, we have the different sections. So the Welcome to the Texas Master Beekeeper Apprentice course. This is everything you need to know for last week's session. And then
Garrett,
for yes.
I'm afraid a lot of us don't see your screen yet. Oh, wait, there's a little piece that says Apprentice Part 2. Here's a little piece showing up now. It's taken a really long time for it to load, I think.
Okay, I might be sharing the wrong screen. So let me fix this quick. Thanks for letting me know. Let's see. Let me share my screen. Okay, can you see it now? Maybe not.
See
Okay. There you go. Does that work now? Can you see the full screen?
Yes, I see it
now.
Perfect,
Thank
perfect.
you.
Okay, so for each week, we have a separate folder for it. So if you go to Classroom, Coursework, for the Welcome to the Master Beekeeper Apprentice course, this is everything for last week. And so we did upload the recordings on here, but if you have troubles watching the recordings from last week, we did make a YouTube page. So you can click on this YouTube page and it'll take you right to all the videos. So nobody's had issues with the YouTube page, so I think maybe we'll start uploading things on YouTube just to save storage. On bottom here, we have our apprentice pretest, and it looks like about 120 people turned the course in. If you have not completed that quiz or that pretest, please take it as soon as possible, just so we can have sort of a baseline. This isn't graded, so we just want to know where everybody's at and sort of their baseline knowledge. And then under the apprentice course from last week, we do have a community notes section. And so one beekeeper, Cassidy, she was taking notes and she thought it would be a great idea to have community notes section. And so we'll start putting all the notes in these community notes sections for each week, just so people can add to it and kind of read up notes from last week. But we'll have this for every single week, and so this will serve as sort of a baseline study guide for everything. But like I mentioned earlier, we'll be making a study guide and a quiz after each of these sessions. And so what I'm really hoping is that after each session that we'll have a study guide and some reading material for you guys to review everything and kind of find things that you should be focusing on. And then for this week, we'll have understanding honeybees. And so once again, we have the community notes section. So if you want to add any notes or any questions in here, that's a really great place for it. We'll also be adding, some of the chat and all the questions asked in the chat in this community notes section, or maybe a different section. But then we had the reading material for this week and last week, understanding honeybees. And so hopefully you guys had a chance to read that beforehand. But if not, it's a good opportunity to kind of read all that information, and it really showcases everything we talked about today. And so if you have any questions about the Google Classroom, I know it's a little bit finicky, so we're trying to work through it, but please let me or Heather know. And some other things, yeah, so quiz afterwards, but somebody had questions about resources. And so I know I talked a lot about resources last week, whether that's with our Ag Life Extension program or whether that's with the Texas Master Beekeeping program. And so after this, I'm going to make sort of a resources tab. right here in the Google Classroom. So you can find where our website is. You can find where all of our free fact sheets are. You can find where the Master Beekeeping Program is. Really, just all of our resources. So hopefully, we can make that available to everybody. Okay, so we had a few questions. So I'm going to answer some of the questions that we had throughout this last week because I think some people might have similar questions. And so one of the questions I had was, what is the difference between a wild and feral colony? And so I had a conversation with this gentleman as part of our course, and he asked me, what is a wild and what is a feral colony? And so when we look at the definition of what a wild colony is, this is sort of an animal or organism that has never been managed or domesticated. Now, for honeybees, they are non-native to the United States, and so they were imported, around the 1600s. And so we really don't have many wild colonies. There may be a few because they could get on a boat or be accidentally shipped to the United States. And so those wild colonies could be established into the United States. But, for us, it's really hard to determine whether a colony is, wild or feral. Now, a feral colony is one that was, managed at some point. And so let's say you have a colony that swarmed and they establish a new colony in a tree or a structure. And so those would be considered a feral colony. Now, typically for me, I don't call a colony feral unless it's been, living unmanaged for at least a year. And so if you have a bee colony that's living in a tree and it's been there for over a year, I'd consider that a feral colony. But it's really hard to know the difference between a wild and feral colony. And I'm not quite sure if we have many true wild colonies here in the US. I know there's quite a few in Africa and Europe, but I'm not quite sure about here in the US. Um, Another question is, are queens mated when you purchase them? And so if you're buying packages or nooks this coming spring, the queens will be mated. And so they come in these small queen cages with sugar. And so as you put those nooks and packages into the colony, the bees will kind of eat that sugar and release the queen. But that queen's already mated. And we'll talk about that a lot more next week when we talk about getting started in beekeeping. One option beekeepers have is buying queen cells. So these are queens that are capped but haven't emerged or mated yet. And so most small scale beekeepers aren't buying queen cells, but if you do buy queen cells, then those are queens that aren't mated and will eventually mate in your yard or your apiary. And like I said, we'll talk about that a lot more next week. Another question was about urban beekeeping. And so I know a lot of beekeepers that attending this course or the advanced course are keeping bees in an urban area, whether that's Dallas, Fort Worth, Houston, or San Antonio. And with urban beekeeping, we'll talk about there's some different things to consider with urban beekeeping because there's maybe more pesticides applied to somebody's home or a homeowners association. There can be less foraging resources for bees. And so I'll talk about that a little bit more in the next coming weeks about the differences between urban and rural beekeeping because there are some differences and things to definitely consider. But if you're interested in learning just about urban beekeeping, I'll be speaking about this for about an hour at the Brennan Bee School, the Central Texas Bee School, in the first Saturday of March. And so if you're interested in attending that course, it's also great to attend that conference because there's a lot of different topics, a lot of different courses, and they're typically taught by beekeepers across the state. And so I highly recommend attending the bee school. I think they typically have somewhere between 700 and 800 beekeepers across the state. So it's definitely one of the largest beekeeping events in the entire country. And so I'll probably have Heather post a link to the bee school if you're interested in attending. Like I said, it's in Brenham and it's a really fantastic program. Also, we will having the Texas Master Beekeeper testing in Brenham, Texas, the day before the event, that Friday. And so, if you do have a year of experience and you want to take the apprentice level exam, you're more than welcome to take the exam in Brenham. And like I said, because you guys are registered for this 10-week course, you can take the exam then. But if you don't have a year of experience yet and you're about to get B's, you can't take the exam yet. And we'll be scheduling some testing events later on this year. And then another question I had was, I've had a lot of people talk to me about some of the recent colony losses, and because I know we've been hearing a lot about this in the news, whether that's social media or even some recent news reports. And so I just want to touch on the colony losses a little bit more. And so here's what we know. We know that beekeepers, especially commercial beekeepers, are experiencing these massive colony losses, and we're seeing these losses happening in California. Now, this isn't to say that these losses are a California-specific issue because bees across the entire country are shipped to California for pollination, primarily almond pollination. And so this is where beekeepers are seeing these dead colonies and these high colony losses. But these colonies are coming from all across, the nation. And so what we did, we were working with Project Apis mellifera, a nonprofit. We're also working with the USDA and other state organizations to, really gather more information. And so we've been surveying beekeepers, commercial beekeepers, to figure out one, what are their colony losses, and what is the breadth of this issue? Like, how big is this issue? Two, we're seeing where these beekeepers are coming from. And so, is this a regional issue or is this sort of a widespread issue? So, for example, are beekeepers that are migrating from North Dakota having more issues, or are beekeepers from Texas having more issues? And so, could be a regional specific issue. And three, we had a team of folks from the USDA go out to California and collect samples to try to diagnose any of the issues and maybe figure it out. Here at Texas, with our extension program, we're working with beekeepers that are shipping bees back to Texas, whether those are dead outs or live colonies, to really figure out what's happening. And so what we're doing is we're working with the Texas Veterinary Diagnostic Lab to do some viral testing and some wax pesticide testing. And so eventually, this diagnostic lab will be available to all beekeepers in the state of Texas. And so if you are interested in diagnostics, we'll have a facility in the state of Texas. And so that might take a little bit longer, but we're still kind of getting that diagnostic lab set up and having some available testing for beekeepers. But in the meantime, we'll be testing them for any known viruses, but even new and emerging viruses. And so, as we'll talk about in the disease section, there's a lot of extremely virulent viruses associated with bees, and they can cause high colony losses. And the problem with viruses is we don't have vaccines to treat or manage viruses. And so if they get out of control within the colony, they can cause colony crashes and lead to, these massive die-offs. And so we're looking at, whether those losses may be associated with virus that we know about, but maybe there's a new emergent virus that we're not testing for. We're also looking at for any pesticide residues in the wax. And so maybe these losses are due to pesticides. And so we're looking at that. And then finally, we're looking at, varroa mite resistance. And so as we get into the varroa section and the disease management section, we'll talk about how to manage your varroa levels. But one treatment that a lot of beekeepers have been using for the past 20, 25 years is amitraz. And so if you use apivar strips, the active ingredient in the apivar is amitraz. And we've been seeing resistance to amitraz for really the past 10, 15 years. So this isn't a new problem, but it's been getting progressively worse over the past 5 years, especially. And so I did some surveying in Texas a couple years ago, and for me, it was really concerning to see how high the amitraz resistance was. And so what we're doing is we're collecting dead mites from the dead colonies, and then we're testing them for whether they're resistant or not resistant to amitraz. Unfortunately, we are seeing pretty widespread amitraz resistance, and so beekeepers are depending on amitraz, then their mites are resistant to it. And so that means there could be more mites in the colony, more viruses in the colony, and that could be one of the major drivers for colony losses. And so, but with honeybee losses, and I'll talk about this a little bit later on, but there's really five P's associated with colony losses. We have parasites, so varroa. We have two pathogens, which are viruses and bacterial pathogens. We have three pesticides. We have four poor nutrition, and then five poor queens. You can also add in poor weather because weather was really variable this last year, but unfortunately, there's probably not one silver bullet. And so even though, mites may be resistant to amitraz and may have higher virus levels in their colonies, if there's less nutrition available or poor weather or the bees are exposed to, pesticides, that could make the mites and the virus problem even worse. And so it's probably likely a combination of all these different factors. And that's one thing as you'll get into beekeeping is it gets more and more difficult. But one thing I'll say is because I think a lot of beekeepers that are attending this course, either you're getting bees or you already have bees, but most likely you guys are probably small scale beekeepers. I know some are maybe closer to Sideliner, where they have closer to 100 colonies, but probably most beekeepers that are listening to this chat probably have less than 10 colonies. And what we're seeing is it's mostly affecting the commercial beekeepers and not as much the small scale beekeepers. But we still need to gather more information about colony losses for the small scale beekeepers. And so Texas A&M, along with Auburn universities and the apiary inspectors across the state, we're launching a survey in April and it ends in May. And so I'd love for all the beekeepers that are attending this course, but also beekeepers across the state to take the survey just to see if this issue is not limited to just commercial beekeepers, but even small scale beekeepers across the state of Texas. And so once that survey launches, I'll be sending you guys a link, and it's really great for me to gather more information because I see know the extent of this problem. Is it just commercial beekeepers? Is it a regional problem? And is it also impacting the small scale beekeepers? But it also lets me know about management practices beekeepers are using across the state and things I can do to help, teach and educate and really resolve some of these issues. And so as I gather more information and as the nation gathers more information, I'll keep you guys up to date. But I hope that's sort of a quick summary about it. Fortunately, it's typically not one issue; it's a combination of all these different issues, and we really don't know if it's regional specific or if it's just targeting commercial beekeepers. But we're gathering more and more information to try to figure out what's happening and why is it happening. And I think that's all the other questions. I think one thing there was a few questions in the chat. One was about, should you buy, nooks or packages from different sources, especially if you are starting from two colonies. And first off, I do recommend if you are starting your apiary to have at least two colonies. Because if one colony crashes or gets really small, you can use that other colony to help that colony out. Or if one crashes, you can split that colony in half and make up your numbers again. So I highly recommend to all beekeepers to start with at least 2 to 3 colonies. And if you're a new beekeeper, it's really fun to try stocks from different beekeepers. They have different selection practices. They're producing queens in different regions, and so you can try out queens from different beekeepers and see which one's best. And I think that's all the questions. And I think as you go, just put your question in the chat. I'll be talking during the presentation, but Heather will be answering questions, and maybe other beekeepers in the room can also answer some of the questions. But if you have any questions that you want, me to address more broadly next week, just shoot me an email, put in the chat, and then we can talk about this at the beginning of each session. Also, we can open up the course at 8 or 8:30, just have maybe like a coffee hour just to answer any questions. And so, beekeepers are interested in starting the course earlier. We can do that just to answer general questions and then start the actual course at around 9 o'clock. Okay, perfect. So I think we'll get started. Okay, let me know if you can see this screen. Okay, should be. Okay, perfect.
Yes.
Okay, perfect. Thank you. I need. Ooh, I'm gonna close my door quick and I'll be right back.
While he's doing that, John, what's your email? Because I'm trying to find it. John?
Okay, yeah. If John, if you're available and want to send your email, just let Heather know and then we can go from there. Okay, so So last week we started our lecture on honeybee biology. I sent you the reading material yesterday, and so this covers both last week's session and also this week's session. And like I said, there's a lot to cover with understanding honeybees. We have everything from the colony structure, wild, feral, and managed colonies. We have a lot of different behaviors and caste, and then we'll talk about physiology. So there's a lot of information that goes with understanding honeybees, and you know, a lot of this stuff may not be on the quiz, and so we'll have a study guide after this session. But I think it's really good to talk about some of these topics because, honeybees are really fascinating, and especially for me, and they differ from humans and other organisms in a lot of different ways because they're an insect rather than a mammal. And so, at the end, I'll talk a little bit more about their physiology and, how they breathe and how their blood works and the nervous system, for example. And so it's kind of fun for me to talk about these different topics because, they are different from, mammals and humans. And it's fascinating to see how, bees function versus, other mammals and organisms. But what's also cool about honeybees is we like to call them a superorganism. And so what that means is there can be somewhere between 5 to 20,000 bees in a colony, and even though each bee kind of has their own function or their own task, they kind of function as a group to make sure the colony grows and survives and collects the necessary nutrients. And so, a lot of ways, all bees are collectively working together as one single group, and we like to call that a superorganism. And so, I'll talk about that a little bit more because that functionality really allows them to live in any part of the world and allows them to survive in a lot of different places and regions. But I think so. So last week we finished with the queen biology section, and this week we'll talk about drones or male biology. We'll talk about worker biology, and then we'll talk about the social behaviors, especially some of their social immunity, because one way that bees and workers combat these diseases is through these social behaviors. And then we'll finish on honeybee physiology. Okay, so drones are the males within the colony. And I think for a lot of beekeepers, we forget about the males, because they're kind of mooching out the workers for food, nutrients, and warmth. They don't perform any tasks within the colony, right? They're not defending, they're not foraging. But even so, they're extremely important because these are the males that are flying out, mating with unmated males, and passing that colony's genetics onto a future colony and onto the next generation. And so, even though within a colony they may seem, I guess, kind of useless, they're really important and they're sort of a way for the genetics of the queen and the colony to go on to future colonies. So, why are drones important? And there's really four major reasons why drones are important. So, number one is the colony genetics. So, within a colony, 50% of the genetics are from the queen because the queen is the only individual that's laying eggs, and she's laying about 1,000, 2,000 eggs per day during maximum colony size. And so most of the genetics are coming from the queen, but the other half of the genetics are coming directly from the males. And like I mentioned early on from last week's session, queens will mate with somewhere between 15 to 20 males. And so those 15 to 20 males are contributing to genetics within the colony. And so really within the colony, there's about 15 to 20 subfamilies within the colony. So depending on the males that she's mating with. And so these subfamilies can have different behaviors and different functions, but this is all to say that the drones are contributing to genetics in the colony, and that directly impacts the colony's honey production, impacts their resistance to viruses and, varroa, and their tolerance to other things. But, one thing that are also important too is sperm production, because the queen will fly out and mate with these males, and she'll store that sperm for somewhere between 1 to 4 years and use that sperm to produce fertilized eggs. So, but if the male isn't producing high quality sperm or high amounts of sperm, that can directly impact how much the queen can store sperm, but can also impact how long the queen can actually store that sperm. So, if the drone is producing really crappy sperm, for example, then the queen might not be able to store that sperm for 2 to 4 years and may die within her spermatheca. And so, the drone's ability to produce sperm and also high quality sperm is extremely important. And then we have just the colony level traits. And so these colony level traits and genetics really go hand in hand. So if the colony has certain genetics from the males within the colony, they're more likely to have these certain traits. And so as a beekeeper, whether you're a first year beekeeper or whether you're keeping bees for, over 20 years, there's certain traits that you want as a beekeeper. So one is you want colonies to produce a lot of honey because a lot of people get into beekeeping for honey production. You want colonies to grow and survive. So they're stronger throughout the season so they can produce more honey. So their ability to grow and get larger is one thing that's really valuable to beekeepers. And then their ability to combat some of these stressors associated with colony losses is extremely important. So, mite resistance, disease resistance, pathogen resistance. A lot of this resistance is directly coming from the male. And so, a lot of these traits are coming from the male and are directly impacting the male. And then finally, queen quality. So queen quality kind of goes hand in hand with sperm production. So if queens are mating with better males, then that'll improve queen quality and queen longevity. If they aren't, queens are more likely to fail and crash at the end of the season. And so drones are really important, even though we view them as sort of a nuisance within the colony. So when we talk about why queen drones are important, number one is queen failure. And so one thing we've been seeing really for the past 15 to 20 years is queen failure. And if you talk to any beekeepers that were keeping bees, 20, 30 years ago, they were talking about how queens would live, somewhere between 4 to 7 years. And so queens used to live a very long time. And now we're seeing that queens are failing within 6 months. And so a lot of research and effort has been put into queens because, queens are failing. It must be a queen issue. But one thing we're starting to realize, and this is some of the research I did during my PhD at Purdue, is maybe it's not necessarily a queen issue. Maybe it's partially a drone issue because if queens are mating with poor quality drones that are producing poor quality sperm, that can directly impact the queen, her pheromones, and her ability to lay fertilized eggs for a long period of time. And so there was a researcher at the Beltsville Bee Lab in Maryland, and he was hearing these reports about queen failure around 2015, 2016, and he was interested in whether drones were contributing to some of these queen failures. And so what he did was he pulled up the spermatheca, and I talked about this last week, but it's an organ that the queen uses to store sperm. And so if you pull out that spermatheca, you can measure what percentage of the sperm is alive versus dead. And queens are remarkable in that they can store that sperm at about 90 to 100% alive for long periods of time. And so most queens should be storing sperm that's around this 80 to 100% alive. And so they're really remarkable at doing that. And if you actually compare them to other bee species like bumblebees or other solitary pollinators, the queens can only store that sperm about 50% alive. So queen, especially honeybee queens, are really good at storing that sperm. And so this was an ARS facility. So this was the control at their bee lab. And so as you can see, the sperm was somewhere between 90 to 100% alive, which is what most queens should be doing. But then queens that were failing in the East Coast and the West Coast, these queens were storing sperm that was about 50 to 60% dead or alive. And so about half that sperm was actually dead that the queen was actually storing versus also requeened. So ones that where the colony was allowed to produce and make a new queen, they were only storing sperm that was 50% live, but there's really variable. But this all say that this could be a queen issue. So maybe it's a queen's ability to store sperm. So maybe she's stressed from pesticides, maybe it's an environment, lack of nutrition. But this could be a drone issue. So maybe the queens are mating with low quality drones, and that's leading to queen failure. But this all say that drones are really important in that they contribute to queen quality and may lead to queen failure. Second is drones contribute directly to colony success. And the reason for that is that they contribute about 50% of the genetics within a colony. And so, I outlined all the traits that you may be interested in as a beekeeper, but a lot of those traits are directly from the males or the drones. And so if we look at mite resistance, and we'll talk about that a little bit more and the traits that bees have to deal and combat mites, a lot of these traits and genetics are directly from the males. We've been working on, how to measure and select for virus resistance. And like I mentioned, we can't treat or vaccinate for viruses. And so the best way is to kind of breed for resistance if possible or manage the mite levels. And we're seeing that virus resistance in some colonies are due to the males. Productivity, different landscapes. And so I think we talk a lot about locally adapted stocks. And Texas is a big state, and there's quite a few ecoregions across the state of Texas. And so some colonies might perform better in West Texas versus East Texas versus South Texas. And one way that colonies can become locally adapted and perform better in different parts of Texas is directly from the males. And then just colony performance. So the colony's ability to grow rapidly in the spring, the, their ability to produce large amounts of honey is directly from the male. So this is really all to say that, drones are important for queen quality, but they're also important for all these traits that you may see as a new beekeeper or even an experienced beekeeper. Okay, so what makes a successful drone? So when I first started looking into drones during my PhD and some of my graduate studies, there wasn't a lot of information on drones. I think a lot of researchers and beekeepers focus on queens, and so we really didn't know what makes a successful drone and what makes a low quality drone. So one thing I outlined in this review, and I'm happy to send this to you afterwards, is there's really three major points that make a high versus low quality drone. And so point number one is sperm production. Number two is sexual maturity, and three is mating behavior. So the drone's ability to track and mate with the queen. Now, for development for a drone, they take 24 days to develop. So that means from egg all the way to when they become an adult. Whereas a worker takes 21 days and a queen takes 16 days. So males or drones take quite a bit longer than the rest of the bees to develop from an egg to an adult. Now, the egg hatches at the same number of days. So for a drone, a worker, or a queen, that egg takes 3 days to hatch. But the development for the larval stage and the pupa stage is longer for drones. But what's really important for this development is sperm production. So drones can only produce sperm during a really short 2 to 3 day window during development. And after that, drones can never produce sperm ever again. And so if something happens during that developmental stage and when drones are producing sperm, that can really cause irreparable damage to the drone. And if that happens, they can never produce sperm ever again. And that could potentially lead to sterile or infertile drones. And if we actually compare drones or males in bumblebees or other closely related bee species, those males produce sperm continuously when they become adults, whereas for honeybees, it's restricted to a really short day period. And so that's what makes, honeybees really unique compared to other bee species. Now, once drones emerge as an adult, they still have to reach sexual maturity. So sexual maturity is when the drone's sperm is mature and able to be transferred or inseminated to a queen. So for sexual maturity, the drones basically stay in the brood box where the young nurse bees are. And they do that because the drones are getting constant care by the workers. So the workers are, sharing pheromones, they're sharing food, they're sharing warmth, and they're really helping the drones reach sexual maturity. And if you actually take these drones and put them into an incubator, they'll likely die within a couple days. And the reason for that is they're highly dependent on the workers for everything. And so I like to call this really the teenage years for drones because they really need the parents and the workers to really take care of them. But this process is really important because even though drones produce sperm during development, the sperm has to move and become mature to be used for insemination. Now, after drones reach sexual maturity, the workers are done taking care of them, and they kick the drones to wherever the honey is stored. And so that's typically on the outskirts of the brood box or up into the honey supers. And the reason for that is the drones are switching from a high protein diet provided to workers to a high sugary diet for their mating flights because after they reach maturity, their goal really is to find a queen and mate with a queen. And so these drones may be going on maybe somewhere between 8 to 10 flights per day. These flights can last around 30 minutes and they could potentially be flying, multiple miles. And so they need that sugar from the honey to sustain these flights, find a queen, and mate with a queen. And so the mating can last somewhere between, 14 days to I've seen reports to even 28 days. And so really the drones are either finding a queen and mating with a queen, or they're dying without successfully mating with a queen. But really what makes a successful drone is these three major points: sperm production during development, sexual maturity during early adulthood, and then mating behavior once they reach maturity. And so this whole process takes somewhere between 34 to 54 days. versus a queen, their entire lifespan's 2 to 4 years. And so their lifespan's quite a bit shorter than queens. Okay, so point number one is sperm is produced during development. And so like I talked about, last stage, honeybees go through holometabolous development. So what that means is they're reorganizing tissues and shifting from a larval stage all the way into a pupa or adult stage. So they're basically switching forms. And so when an egg hatches, the larva is really small, about the size of a grain of rice, and that larva gets bigger and bigger each day, and they go through these about 5 molts. So as that larva gets bigger and bigger and bigger, their testes, which is where the sperm is produced, start getting bigger also. But then once it reaches fifth instar or their final molt, and this is when the drone larvae are really large and almost overflowing in the cell. And so it almost seems like the drones can't fit in the cell. And for new beekeepers, you won't see this until maybe May or June, but you'll see the drone cells and you'll see the really large larvae inside the cells. And this stage is really important because this is the first stage of sperm production. And we call this spermatogenesis number one. So what that means is they're producing these sperm cells and producing enough sperm cells for fertilization and mating afterwards. And then once the cell is capped and the larva spin a cocoon into this prepupa stage, this is spermatogenesis stage number two. So they're basically producing sperm during this 2 to 3 day window from this fifth instar larva into this prepupa stage. And so after this, drones can never produce sperm ever again. So this stage is extremely important. And one thing I'll talk about a little bit more, especially during the mite management section and maybe some of the mating biology section, is that drones, because they develop longer, they have more fat in their tissues, they're really mite magnets. And so mites like to jump in and feed on the males. And so the mites are feeding on the males that can impact their sperm production and their sperm quality. And so that's why it's extremely important to manage your viral levels because that can directly impact the males that future queens are mating with. And so also with queens or drones, once they reach this pupa stage around the purple-eyed, pink-eyed stage, this is when the sperm tail is formed. And so the sperm heads are formed during the larva and prepupa stage, but then the tails are formed once they reach the pupa stage. And so the sperm tail is really important for sperm swimming, so during sexual maturity, but it's also important for that sperm to have these tails to swim into that spermatheca or the queen's sperm storage organ. So here's a little bit more about an adult male anatomy. And so this is what the anatomy looks like. And so this is what the testes is. So this is where the sperm is produced and stored. But in order for the drone to mate, the sperm has to successfully swim from this testes into this organ called the seminal vesicles. So if all the sperm is still in the testes and a drone mates with a queen, that cannot be transferred to the queen. So the only way that sperm can be transferred to the queen is if that sperm's actually in this organ called the seminal vesicles. And so that's why the sperm tail is really important because it allows the sperm to swim from one organ to the next to be used during mating. And then we have these mucus glands. And so I talked about this a lot in the reading materials, but these mucus glands produce, hundreds of different proteins. And these proteins do a lot of different things. One, these proteins protect the sperm. And so they protect it from, free radicals or things that could damage the sperm. They provide nutrients to the sperm to keep them alive for longer periods of time. They help the sperm, live inside the queen. So some of these proteins are actually inseminated with the sperm to keep them alive in the queen for longer periods of time. A lot of these proteins serve as antioxidants. Some are antimicrobial. So this is all to say that there's a lot of proteins in this mucus glands, and they have a lot of different roles for the drones. And then we have the ejaculatory duct. And so during the mating process, the sperm is transferred from the seminal vesicles through the ejaculatory duct into the queen's oviduct. And so the sperm during insemination will travel from the seminal vesicles through the ejaculatory duct into the queen. And as I'll show, this whole organ, like the bulb and the ejaculatory duct, will basically avert outside the drone. And so this penis will stick out, and that's when inseminated into the queen. But what's also cool about this is there's these horns. So there's about 2 horns on the back of the drone, and these horns basically go into the queen and keep the drone basically locked into the queen during the actual insemination process. And so because drones and queens are mating with the air, this allows the drones and queens to stay attached even if they're falling out of the air and allow that insemination process to actually occur. And so these horns are extremely important for the actual mating process. Okay, so now talk a little bit more about sexual maturity. So once the drones emerge as adults, they stay in the brood box. And so, if you're a new beekeeper, you're buying these deep boxes. That's where the queen's laying eggs. That's where the brood is, and that's where the young nurse bees are. And that's where the drones immediately go after emerging as adults. And they do that because they're receiving constant care by the workers. So the workers are providing food, they're providing warmth, they're basically providing everything the drone needs to survive, but also reach sexual maturity. Now, what is sexual maturity for a drone? It basically means your ability for that sperm to swim from the testes to this organ called the seminal vesicles, because once that sperm's in the seminal vesicles, it can be transferred and inseminated to the queen. So the workers are helping the drones reach sexual maturity and get that sperm into the seminal vesicles. Now, this is what an immature drone versus a mature drone looks like. And so if you take a drone and you squeeze them, their endophallus will pop up. So basically, this whole organ will pop out and be everted outside the drone. And this is the part that actually goes into the queen, and this is the part that actually inseminates the queen. And here's the horns right here. So these horns go inside the queen and make sure that the drone is locked into the queen during insemination. Now, if you see a drone that looks like this, where it's very clear and there's no white or brown spot at the tip of this endophallus, then that's a sign of an immature drone. But if a drone looks like this, where you can see some white, which is the mucus proteins, and the brown, which is the sperm, this is a sign of a mature drone. So once your colonies begin producing drones, and actually our colonies started producing drones last week, so we actually have some drones producing in our colonies. And so probably within the next month or so, you'll probably have some drones within your colonies. If you want to check if they're mature or not, you just take them and you squeeze them and this endophallus will pop up and you can tell whether they're mature or immature. But in order for a drone to reach mature, it takes about, 48 to 50 days because the drones need to go through 24 days of development plus another 14 or so days to reach maturity. So if you see some, drone eggs in your colony, it'll take about, 50 days for that drone to actually reach maturity. And then finally, the drones, once they reach maturity, need to go out, find a queen, and mate with a queen. So, like I mentioned earlier, the drones are basically pushed out from the workers. The workers are done taking care of them. They're done providing resources, and they want to focus on, younger and newer drones. And so these drones are kicked out to where the workers are, to where the honey is stored. And so if you're running colonies in single deeps, so one deep box, then that's typically where the honey is stored or where you're feeding. So on the outermost frames of the boxes. But if you have honey supers on, most of the time the mature drones will be where the honey supers are. So if you have drones in your honey supers and you squeeze them, about 70 to 80% of these drones will be sexually mature. Now, the role for a drone once they reach maturity is to basically find, locate, and mate with a queen. And to do that, they go to these areas known as drone congregation areas or DCAs. Now, these DCAs are basically, I guess you can call them kind of a bar, kind of a bar for these drones because about 1,000, 20,000 drones basically congregate in these areas and they wait for a queen to arrive. If a queen doesn't arrive, they fly back, refuel, and then go back to these mating areas. And so it's basically a way for drones to wait for a queen instead of flying randomly to find queens in the air. And so we still don't know much about why and how drones find these DCAs. It's probably partially landscape. So drones may use certain landscape cues, such as a forest or a valley, to find these DCAs. They may also use pheromones. So they may make a pheromone track from the colony to the DCA to attract more drones. And so we're still learning more about why drones and how drones form these DCAs, but it's a really fascinating behavior. Now, let's say a queen does arrive at those DCAs, the drones will form these massive drone comets and chase after the queen. And if there's 1,000, 20,000 drones competing for one queen, you can imagine how competitive that environment is. And only about 15 to 20 males are actually successful at mating drones out of the 1,000, 25,000. And so it can be extremely competitive environment, and only only a few drones are actually successful at mating with a queen. And actually, I'll go back here. So after a drone mates, they immediately die. So their entire reproductive tract is ripped out, and then they, fall and die to the ground. And so drones can only mate once, and after they mate, they're basically done. So what causes a poor drone, or what causes also a high quality drone, is sperm production during development. Two, their ability to reach sexual maturity, and three, mating behavior. So ability to find, locate, and mate with a queen. So what can cause a poor drones? And this can be the same with queens. So what causes poor queens and poor drones? Number one is environmental factors. So, poor nutrition. So colonies require, good nutrition, whether that's pollen or nectar, to sustain making drones and producing high quality drones. And so if your colony isn't bringing in a lot of pollen or nectar and they're reaching what we like to call a pollen dearth, then that could be a sign of poor nutrition in your colony, and that can lead to not only less drones, but poor quality drones altogether. And so if you're a beekeeper raising queens, and a lot of you might not be, but if you are raising a few queens within your colony, it's really important to have, good nutrition for that queen, but also the drones within the colony, because these are the drones the queen's mating with. Pest pathogens and diseases. So, as I'll mention in the disease section, a lot of these pathogens and diseases are really targeting the developmental stage. And like I mentioned earlier, this is when the sperm is produced for the drones. So, if the parasitic mites are jumping into the cells, feeding on the drones, they're transmitting viruses or pathogens that could directly impact the drones and cause them to be sterile or infertile altogether. And so managing your viral levels and managing your pathogen levels is really important, because if you don't, that can lead to not only poor quality queens, but also poor quality drones. And then three, miticides. So I'll talk about miticides a little bit more in the rural management section, but beekeepers use a wide range of miticides to kill and manage their rural mite levels. And some of these range from highly synthetic miticides to more organic miticides. Now, all these miticides have some effect on the drones, but some can have harsher effects on the drones. So, some of the synthetic miticides, such as, apivar strips, can drop sperm quality by 50%. And so instead of a drone producing 90% live sperm that's high quality, it can drop that sperm quality to 50%. And so miticides have a huge effect. But even oxalic acid, formic acid, and some of these organic options can also impact and drop the sperm quality. So just be aware that if you are applying miticides, you are going to be impacting the drones. As a new beekeeper, your queens could be mating with any drone because you're mating in the air. So it's even if you manage your drone levels, your queen's still gonna be mating with drones from your neighbors that aren't managing them properly. So it's kind of tough to manage, but these are just some things that can impact the drones. For pesticides, so there's a wide range of pesticides used. We have insecticides, we have fungicides, even herbicides, and these all can impact drones and sperm quality. And then finally, temperature. So extreme temperature and extreme temperature waves, which is definitely a thing in Texas. It can be 110 to 120 degrees. And if these extreme temperatures occur and the drones are actively flying, that can not only impact their sperm quality, but their ability to mate. And so I know this last summer I bought stocks from across the country, and some stocks that were, raised and adapted to the Midwest didn't perform as well here in Texas. And I actually saw some drones that were flying and then they started falling from the ground because it was just so warm here. So this is all to say that, if you're buying, stocks from Texas, these drones may be more adapted to the extreme temperatures in Texas and might be able to find a queen or find a queen to mate with. But if you're buying stocks from different parts of the country, those drones may be flying, but might not be able to, live and fly in these extreme temperatures. And so there's a lot of environmental factors, but like I mentioned, you can manage your drone population as well as you can, but your queens are still gonna be mating with drones from other colonies because, typically your queens are gonna try to find an unrelated drone to mate with, so we don't have any inbreeding. So you can manage your drones all you want, but if your neighbor isn't, then that could impact your queen quality and the sperm quality of the drones. And then, we also have these genetic factors. So as a new beekeeper, and we had this question in the chat was, should I buy nooks and packages from different producers? And the thing about, buying bees from different beekeepers is that they're maintaining different genetic lines. And so, we talk about Italians, we talk about Carniolans, we talk about specific stocks such as Poline, Russian. There's a lot of different breeding lines available, and they're all genetically different. And because they're genetically different, they can produce different types and quality of drones. And so, this also impact the queens and some other traits, but it's kind of hard to know until you buy them. Beekeepers could be marketing their stock as one thing, but they could also be producing lower quality drones. So it's one thing that you really can't control too much, but it's something to be aware of that if you're buying stocks from different sources, that they could be producing higher versus low quality drones, especially here in Texas, which is different from other parts of the country. Okay, so the first part I talked about, sperm quality, and then talked about, sexual maturity, but next I'll talk a little bit more about mating behavior. So like I said, the drones, once they reach sexual maturity, will fly to these drone congregation areas called DCAs, and they have somewhere between 1,000 and 25,000 males. Now, the average drone will fly somewhere between maybe 2 to 10 flights per day, and these flights can last around 30 minutes. They're typically leaving the colony in the afternoon, so around 2 o'clock to 6 o'clock, but kind of depends on how hot it is outside and whether it's raining and other environmental factors. But typically, though, they'll fly, in the afternoon. And so if you see, a bunch of drones flying, those are typically mature drones going on mating flights and finding a queen. And so they basically sit at these mating areas and they wait for a queen to arrive. If a queen arrives, they form these massive comets and chase after the drone, and a lucky number will actually successfully mate with the drone and inseminate the drone. And I showed the same video last week, but I'm going to show it again because I think it really illustrates the mating behavior of queens and drones. So here's a queen going on its mating flight, so a virgin or unmated queen. So this queen's also going to a drone congregation area because they want to find drones to mate with and 15 to 20 drones to mate with. So this is DCA. You see all these drones flying, and now they're forming this massive comet and chasing after the queen. Now they're kind of knocking each other away because they want to mate with the queen. But one of these drones will be lucky enough to latch onto a queen, and they latch onto the queen. They put their horns into the oviduct so they can latch onto the queen and stay attached. And then they avert their whole reproductive tract into the queen and start the insemination process. Now, once they're latched on, they're basically paralyzed and fall in the air during insemination. And then once the mating process is over, the drone will basically fall and then die afterwards. So drones can only mate once during their lifespan. And then the queen will go on and mate with more and more males. So a little bit more about the mating behavior and some of these drone comets. This was a really cool study done in Puerto Rico. And one way they tracked drones and these DCAs was through these small radio tracking antennas. And so they put these antennas on the back of a bunch of drones across the island of Puerto Rico. And by triangulating where the drones are flying, they could see not only how fast the drones are flying, but where they were mating at. And so these small triangles are the different apiaries where they track the drones. And so you can see there's maybe 30 to 40 different colonies that they track drones from. But yet the drones are only going to maybe 4 to 5 of these drone congregation areas. And so even though there's a lot of these apiaries, they're only going to a few of these DCAs. Now, what's interesting is when the drones were really young and first started going on their mating flights, they did sort of orientation flights. So kind of like scout bees, where they were going to multiple DCAs when they're really young, and trying to find maybe the sexiest area. And then once they found one that they really liked, they went back and forth between that one DCA. And so, for example, a drone may be marked from this location and maybe go to one of 5 of these different DCAs. And then once they get older, they go back and forth between one DCA. And one reason for that is because they need to go back and forth between one because they don't have much time left. They need to find a queen to mate with because if they don't, they'll eventually die without, passing genetics and passing their sperm to a queen. And so things become a bit more frenetic after that. But this all say that, drones typically congregate in, 1 or 2 areas in a local area. Here's some data that I collected about these different lines. So these are different breeding lines that you can buy as a beekeeper. So some are Italian, Carniolan, some are part of a breeding program, but this is to say that they have different flight behavior based on the line. And so this is the age at first flights when drones begin their first flight. So for this line, they begin their first flight around 8 days. For lines 2, 3, and 4, they started their flight around 6 days. So that means that they're reaching sexual maturity for the first 6 to 8 days of their lifespan. Once they reach maturity, they begin flying. And so this typically is occurring around 6 or 8 days. Now, as for the number of flights per day, line 1, they're taking about 10 flights per day. Line 2, about 8 flights per day. And then 3 and 4, about 6 flights per day. So the average drone is taking around 6 to 8 flights per day. And some of these flights can last somewhere between 10 minutes to even 40 minutes to an hour. So some of these flights are extremely long, but the average is 30 minutes because the drone doesn't want to stay outside the colony too long because if they do, they risk not only predation from like a bird or something else, or they risk running out of fuel or honey to make it back into the colony. And so typically a drone will fly only for about 30 minutes, come back, refuel, and go on another flight. But this really depends on the weather. So if it's really cloudy outside, if it's raining outside, these flights will be a lot less. So, but if it's a really perfect day and the weather is, 70 to 90 degrees, the drones will be taking as many flights as possible. Okay, so the first part was about drones, and actually one thing I want to mention about drones because I didn't have a slide about this is that a colony typically invests into drones when they have a lot of resources to sustain colony growth and colony survival. And so once a colony has enough resources available to, grow and survive, they start investing into the drones. So the drones are basically, flying sperm. And so once once your colony reaches the summer or the spring and they're bringing a lot of pollen or nectar, you'll start seeing a lot more drones within your colony. Now, there's only about 100 to 500 drones within a colony at a given time. So, if you compare that to 10 to 20,000 bees, the drones make up such a small percentage of the actual colony. Once the winter hits, all the drones are evicted and killed off. And so you may have a few drones in your winter clusters, but most of the time the drones are kicked off and killed. And so you won't see drones in your colony during the winter because they're taking up a lot of resources that are necessary to grow and survive throughout the winter. And so drones are taking a lot of those resources, and that's one reason that they're evicted and culled from the colony. And so our drones, our colony started producing drones this last couple weeks. They'll probably start producing drones, more and more as the spring hits, especially here in East Texas. But it really depends on your region when your colonies begin producing drones. But I'd probably say around March or April. And if you get in colonies around April, your colonies may begin producing drones, in May or June. Okay, so now we're talking about workers. So workers make up the majority of a colony. So, if a colony has somewhere between 5 to 20,000 bees, about 90 to 95% of the bees in a colony will be these workers. And like I mentioned beforehand, they're genetically the same as a queen. And so they're both females and the result of a fertilized egg. But because of changes in development and restriction of food during development, these workers are non-reproductive, so they can't lay eggs in most cases. And they have specialized features that allow them to do all these tasks within a colony. So ranging from nursing behavior to foraging behavior to guarding, all these specialized traits that the colony needs to grow and survive. And so workers, even though they don't get as much love as the queens, they're extremely important for colony survival and growth. So why are workers important? So workers perform all the important tasks within a colony, and so they're really important ranges from everything from keeping the colony clean to collecting resources. And so their major importance is house cleaning. And so some workers, their only task is to go into the cells, clean out any dirt and debris, and really prime that cell for the queen to lay eggs. And so queens really want a clean place to lay eggs. You can imagine as a hospital bed. You don't want to go to a hospital where there's dirt everywhere and mud on the ground. Same with the queen. The queen does not want to lay eggs if the cell is not clean. So some workers will actually go into the cell, clean it out, make sure it doesn't have any diseases or pathogens, and make sure it's primed for the queen to lay eggs. And so this house cleaning is extremely important for keeping not only the cells clean, but the entire colony clean. And then some bees, their only task is this nursing behavior. And so they form these specialized glands in their head that produce this milk or jelly-like substance. And so these nurse bees, their role is to feed the young developing larvae, take care of them and feed them, and make sure that they grow into adulthood. Some of these nurse bees, their job is to attend the queen, groom the queen, and make sure that she's healthy. And some nurse bees, their job is to, take the pheromone from the queen and spread it throughout the colony. And so nurses are really important for all things from taking care of the brood to taking care of the queens and even the drones. Then we have the cell builders. So these are the workers that form specialized glands in their abdomen to produce new wax. And so as a new beekeeper, you'll be buying a lot of plastic foundation. But in order for a queen to lay egg or for the colony to store honey or nectar, that plastic needs to be drawn out into wax. And so these cell builders produce wax. They mold it with hands on their head called mandibles, and they form these hexagonal cells on that plastic foundation. If you're starting from a top bar colony, they won't use that plastic, but they still need to build that wax to build the hexagon of cells to lay for the queen to lay eggs and for the workers to store, nectar and pollen. And then some bees, their only task is to guard. So there's a subset of bees that sit in front of the colony and they basically survey the whole area for any predators or disturbances on the colony. And so let's say there's a disturbance or predator that's gonna likely impact the colony, like a bear or maybe something knocks over the colony. These guards basically produce alarm pheromone that alerts the rest of the bees, and that tells the rest of the bees to come out and defend the colony. And so these guards are really important for, serving the area for any of these disturbances, but also alerting the rest of the colony. And then finally, we have the foragers. So these are the bees that are physically leaving the colony and collecting all the resources the bees need to survive and thrive. So this includes pollen, which is their protein source. This includes nectar, which is their sugar source that they convert into honey. Propolis, which is used as a glue and also antimicrobial properties. And then water, which helps ventilate the entire colony. And so these are really the five major categories of tasks the workers do. And what's really cool about workers is they change over time and they behave over time and shift through all these different tasks. And so one worker will go through all these different tasks through their entire lifespan. And so a worker will go from housekeeping to nursing to cell building all the way to foraging during their entire lifespan. And their whole anatomy and physiology changes to become specialized for each task. So these tasks and how workers do these tasks really depend on the age of the workers. And so when the workers are really young, they do most of the in-hive tasks. So when the workers first emerge, when they're about 1 to 5 days old, they do all the housekeeping tasks. So those are the bees that are going to the cells, cleaning the cells, and removing any dirt, debris, or any diseases. And so these housekeepers are extremely important for keeping the colony clean and hygienic. So they want to make sure it's sort of like a hospital room where it's free of any pathogens, it's free of any debris, so the queen can lay eggs and the young baby brood can really grow and survive. After that, they become nurse bees. So they start building specialized glands in their head to feed and take care of the developing larvae. They also attend the workers. And so basically, they're doing these kind of nursing behaviors to take care of everything from immature drones and workers to queens. And then they become the cell builders. So they start building wax in their glands to produce new wax. And so basically, for the first 10 to 15 days, they're doing these kind of in-hive tasks. And then they shift over to outside tasks. So they start with this garden behavior and then they transition to foraging behavior. But really, once a worker emerges, their lifespan is only about 21 days. And so an average worker, especially in the summertime, will only live 21 days. And they're transitioning from one task to the next. And so one worker will maybe have 10 tasks throughout their entire life. Now, I will say, and I won't talk about this too much, is that worker's lifespan changes once they become summer to a winter bee. And so winter bees, because the queen's not actively in brood, these workers need to live for, 2 to 4 months. And so summer bees, they only live 21 days, whereas winter bees may live somewhere between 2 to 5 months. And so they basically change their whole body to live throughout the entire winter. And so right now your colonies and your bees are probably these winter bees, but they are probably starting to transition into these summer bees. So here is the different tasks. So this was some really cool studies done in the 80s and 90s, and they basically tracked the age of workers and the tasks they performed. And so this white dot is the average age that they perform a task, but the black bar is really the range of ages that they perform a task. And so these are really all the different tasks. One, two, three, four. So each worker goes about 11 or 12 different tasks. So when a worker is around 1 to 10 days old, they'll do the cell cleaning and the capping behavior. Once they get to around maybe 5 to 15 days old, they do the nursing behaviors. So they start building, glands in their head to produce that jelly or milk-like substance for the developing larva. But they're also tending the queen. So they're forming that retinue around the queen, grooming the queen, and then transferring nutrients and resources to the queen. But they're also collecting the pheromones from the queen to spread throughout the colony. Once they get to around 10 days old to 20, they're doing some of the comb building tasks. They're receiving nectar. So some workers, their goal is to collect nectar from the foragers and then convert that into honey, which is stored for longer periods of time. Some are, collecting and packing pollen. So the pollen comes back from the foragers and it's packed and converted into bee bread into these cells. And then some workers, their job is comb building. So, these workers around 15 to 20 days old are doing everything from receiving the nutrients to cleaning the colony to comb building. So really for the first, day to, 20 days, they're doing all these inside tasks, range from cleaning to brood and queen tending to comb building, and then they transition to outside tasks. So this starts with ventilation, so collecting water, ventilating the colony, and making sure that the colony has a very specific temperature and humidity. So colonies and bees are really good at maintaining a colony at 95 degrees. And one way they do that, especially in these extreme temperatures in Texas, is by collecting water and ventilating the colony. And so some workers, their only role is to collect that water and ventilate the colony. Some workers, their role is to sit in front of the colony and guard the colony, and then we have the foragers. And so, each bee will go through these 10 or 12 tasks throughout their lifespan, and this can, last from 21 to upwards to 30 days. Okay, so a little bit more about these housekeepers or nurses. So if you see a frame with a bunch of brood, a bunch of bees, and you see all these young fuzzy bees, these are typically the young housekeepers or nurse bees. And as you can see from this video, some bees are actually going into the cell and cleaning out the cell. And so this bee right here is actually going around here, looking at the cell and making sure that's primed and ready for the queen to lay eggs. Because the queen's laying somewhere between 1,000 and 2,000 eggs per day. And so the workers have to make sure that there's enough cells ready for the queen to lay eggs and for that brood to develop. Some of these workers, are actually going to the cell and basically regurgitating that jelly or milk-like substance to the young brood. And so this bee right here is actually going in there, feeding the brood, but it's also making sure that the brood is healthy and strong. So making sure the temperature is right, making sure it's cared for, and make sure it's groomed. Because these larvae are really sensitive, and the workers make sure that the larvae are able to survive until adulthood. And so these housekeepers and nurses are extremely important for taking care of the brood and make sure the cells are clean and ready for the queen. And then we have the guard bees. So this is a hive entrance. And so some bees that sit out in the front. So some of these are foragers, so they're leaving and coming in and outside the colony. But some of these bees are basically sitting here and getting ready to basically purview the entire area and see if there's any predators or disturbances to the colony. And so if a bear arrives, for example, these guard bees are the first responders. And so they let the rest of the colony know that something's happened to the colony and they need to fight and defend that colony. And so they release a very specific alarm pheromone, and that tells the rest of the colony to come out and try to push away and get rid of that predator. And so the predators, and we'll talk about this a little bit more in the pest section, but they can be, wasps, they can be ants, they can be small hive beetles, they can be, bears or raccoons, they can be humans, for example. But these guards are basically the first responders for the entire colony. And that's one reason that we smoke a colony is to mask that alarm pheromone. So you'll always have a few guard bees in the front, but by applying smoke, you basically mask that alarm pheromone so the rest of workers can't detect that and they won't come out and get stressed out and maybe begin pursuing you. So smoke is really important to mask the alarm pheromone and not alert more of these guard bees. Okay, next we have the foragers. So these are the bees that are actually going out into the field, and these are only bees that actually leave the colony unless there's certain instances such as swarming. But the bees are going to the flower and they're sticking their tongues right here, their proboscis, into the flowers to collect some of the nectar. And indirectly, as they're collecting the nectar, they have these pollen baskets called carbiculates that are collecting some of the pollen. And so a lot of times when bees are collecting the nectar, they're indirectly collecting some of these pollen and bringing that back to the colony. Now, some foragers, their only role is to collect pollen, and so they're the ones actually actively going out and collecting pollen. Some foragers, their only role is to collect water, and some is to collect resin to convert into propolis. And so there's a lot of different foragers, but primarily they're collecting the pollen and nectar that's needed for, colony growth, brood development, and, survival. So, I'll talk a little bit more about pheromones, but one ways that, workers and bees communicate flower sources is through a waggle dance. And so bees do a very specific dance in the colony, and that tells the rest of the bees where that flower source is. And based on how this dance is done, the angle of the dance, and how long this dance is, that tells the workers exactly the direction of that flower, how far away that flower is, and how good that flower resource is. And so this is generally what the waggle dance looks like. And so bees can detect the angle of the flower from the sun. So based on the angle of this dance, it tells the bees the direction of the flower. Based on how long this dance is, right here along the straight line, that tells the bees the distance from the colony to the flower is. And then in the middle, they do this wiggle dance where they shake their butt. And based on how vigorous that wiggle is, that tells the bees how good that resource is. So there's some really cool studies showing that if you put out, I guess, two sugar sources, and one had a higher concentration of sugar and one had a lower concentration of sugar, the bees that are collecting that sugar that has a higher concentration will come back and do a more vigorous dance. And so that's one way that the bees look at the resource and provide that information to future workers. And so they'll do this wiggle dance and they'll repeat it a few times. And other workers that have not been to that flower source will follow this bee around and repeat that wiggle dance. And that's one way they learn about the wiggle dance, but also learn where that flower source is and the quality of that flower source is. And so that's one reason that if you see a bee at one flower 1 minute and come back 5 minutes later, there's hundreds of bees, it's because they're communicating these flower sources through this very specific waggle dance. And these wiggle dances can differ based on how far away the food source is. And so if it's a really short food source, so a couple feet away, they'll do kind of a short circle. But if it's a long wiggle dance, they'll do a wiggle dance that looks just like this. And here's a video of what the wiggle dance looks like. And we like to call this the dance floor. So this bee is doing this wiggle dance on this dance floor, and this worker is trying to learn about where that flower source is. So this worker is following this around, and the worker is constantly remembering and learning about that flower source. So learning about what angle that flower is from the sun, how far away is that a flower source, and they're also learning about how good that flower source is. So based on how vigorous this wiggle is right here, that tells the bees that this is a really great resource and they want to fly out there. And so there could be, two wiggle dances happening at one point. And if they are, the workers may be, following both dances and figuring out which one they want to go to. And so for that study where they had a source with a high quality sugar and a source with a low quality sugar, most of the bees followed that dance to the high quality sugar and went to that source. And so one thing I also want to mention is laying workers. And so in situations where your colony is unable to raise a new queen, and so if your larva reach a certain age, around 3 days, then the colony is unable to raise a queen from that brood cell. And so in that situation, the colony has no chance of raising a new queen. Now, for a beekeeper, you can requeen or add a queen to the colony and make sure that colony's queen right again. But if you don't replace that queen, then eventually some of those workers will become laying workers. Now, like I mentioned, the workers do have ovaries, but they're shriveled compared to a queen. So the ovaries are quite a bit smaller. And so they can't produce very many eggs. They also don't have a spermatheca, so they can't fly out and mate with a drone to store sperm. But workers can lay unfertilized eggs. So that means all these eggs become males. And typically, when you see a laying worker colony, you'll see a lot of eggs within one cell. And because the workers' abdomens are very short, they're unable to get all the way to the bottom like a queen. So you see a lot of these eggs kind of on the side of the cell. You'll see them kind of curve like this rather than one egg right at the bottom as a queen. And so if you see a situation like this, that's a sign that there's a laying worker and some workers in the colony have begun to lay eggs. But like I said, all these eggs will become males, and I think it's just one way for the colony to still pass their genetics to the next generation, even though the colony will likely crash at some point. One thing I will say is there is a certain subspecies of honeybees in South Africa called Apis mellifera capensis. And these ones actually have anarchy worker bees. And so some of these workers are able to produce um, fertilize eggs, but they can't mate. And so what they do is these unfertilized eggs can become queens through a very, very special process. And so, especially in South Africa, you have these anarchy workers that actually go into other colonies and really take over the colony and really take over the queen. It's only found in South Africa, but it's a really, really cool behavior. And I don't have time to talk about it too much, but if you want to look up anarchy bees and look up laying workers, there's a lot of really cool information about that very specific subspecies of bees. And yeah, I think that's everything about workers. Okay, so a little bit more about the anatomy of a worker. So, like all bees, they have really three major parts of a bee. So you have the head, which is where their brain, antennas, and eating parts are. You have the thorax, which is basically their locomotion and their motor. So the legs are on the thorax, and then you have the abdomen, which is where the gut is, where the heart is, where the digestion is, everything is as part of the worker. And so I'll talk about these in a bit more depth, but just for you guys to know, just know what a head is, a thorax, and an abdomen is. So one thing about workers is they have barbed stingers. So that means is if a worker stings you, there's a barb on their stinger that gets stuck into your skin. And so once a worker stings you, this whole gut and stinger is ripped out and the worker immediately dies. Now, queens also have stingers, but queen stingers aren't barbed. So, if queen does sting you, they won't die immediately after stinging you. Other bees, so most bees don't have barbed stingers. I mean, most bees don't have stingers in general, but the bees that do have stingers, like bumblebees, they also aren't barbed, so they can sting you consistently. But just know that workers, if they do sting you, they typically die. Now, as part of a stinger, there's really three major parts. So you have this barb stinger, which is a part that goes into the skin, and that's where the venom is transferred from the worker into whatever they're attacking. We have the venom sac, which is where the venom is actually produced and stored. And then you have muscles along this that pump that venom into your skin. And so if you get stung by a bee, this stinger will go into your skin, and then this pump will pump this venom right into your skin. And so one thing I do recommend if you get stung right away is to try to scratch off, if you see me, but you try to scratch off that stinger, because if you squeeze it and try to pull it out, you're basically pushing that venom more into your skin. And so I always use my fingernail to really scratch off that stinger just so you don't get more venom into your skin. Because if you grab it, it's going to push all that venom and more venom into your skin. Now, the bee venom is a mixture of a lot of different things, and people use it for medicinal purposes. So, certain sting therapy. I know there's benefits for this bee venom for a lot of different diseases. So, I know it's been really useful for Lyme disease. It's been really powerful for other diseases. So, it does have some therapeutic benefit. If you do like getting stung, you can give it a shot. But if you're allergic or if you're not sure if you're allergic, it's probably not the best option. But this venom is a mixture of a lot of different things. So it includes proteins, it includes enzymes, it includes peptides, and they're all designed for causing pain, causing inflammation, and a potential allergic reaction. Because the goal of the worker is to sting whatever is disturbing their colony and try to push them away and get them to leave their colony. But the most powerful part and what makes up 50 to 60% part of the venom is this melatonin. And this melatonin is really what causes allergic reactions and causes swelling and inflammation. And so this melatonin can change over time. So if you get stung by workers in the winter versus summer, for example, the venom has more of this melatonin, and you're more likely to have allergic reaction. And from my experience, it's a lot more painful to get stung in the winter versus the summertime, and it's mainly due to this melatonin. One thing I will say about bees, because I think a lot of people are getting bees and they may be worried about aggression or getting stung. Bees, especially honeybees, don't disturb you unless your colony is disturbed. Now, some lines are more aggressive than other ones, and they are. So every time you get into the colony, and even after you smoke, they're chasing and pursuing after you. I do recommend requeen that colony with more docile and gentler queen. But in most cases, I just wear a veil around our colonies because they're really nice and easy to work with. And so in most cases, bees are really nice and easy to work with and are extremely gentle and docile. Um, and really the only time they disturb us is if something's disturbing their colony. So the colony tips over, their colony is kicked. There's weather outside, so they're stuck in the colony for, multiple days. Or if you mow around the colony. So if you do have a colony, you're setting up a colony, I do not recommend mowing directly around the colony because that will aggravate the bees and cause them to become more aggressive. And so if you do have to mow around the colonies, do wear a bee suit, but remember that they may be aggravated if you directly mow around them. So loud noises like that can really aggravate the bees. And I've actually talked to a lot of arborists that go up into trees where bees are living in tree cavities, and they probably don't notice the bees right away, but the minute they start that chainsaw, that's when the bees become aggressive because of these loud noises and loud vibrations. And as I'll talk about in the physiology section, bees hear through vibrations, and so these heavy vibrations aggravate the bees and disturb the bees. So lawnmowers, chainsaws, any type of loud noises can aggravate the bees. But in most cases, if the bees aren't disturbed, they won't bother you. Now, I can't say the same for yellow jackets, but bees are docile for the most part. Okay. So one thing I really love about honeybees is their social behavior. And I think this is one thing that most people love and really become passionate about honeybees is that, each individual bee, as I talked about early on, are doing individual tasks. And so some are doing nursing, foraging, but they all work together in a social group, in a social collective group for the betterment of the entire colony. And so you think about it this way, the workers basically gave up their entire reproduction, their ability to pass, genetics to the next generation for the betterment of the entire colony. And so they're all working together in what I like to call a social organism. And so they all perform different tasks, but they all kind of work in conjunction for the betterment of the colony, whether that's growth, survival, honey production, and so on. So the superorganism, we like to call honeybees a superorganism because there's 10 to 20,000 bees working together as one. And so some bees may be doing individual behaviors, but they can be working together as a group to have these social behaviors to combat diseases, to groom the colony for hygienic behavior, for all these different things. And so they're collectively working together for the betterment of the entire colony. So one of the really cool social behaviors for honeybees is swarming behavior. So swarm behavior occurs when the colony either becomes overcrowded, so there's not enough space on the colony for the queen to lay eggs, not enough space for the bees to store resources such as nectar and pollen, and not enough space for the colony altogether. And so they may swarm in that situation. If there's a lot of resources coming in, so the bees are bringing in a lot of nectar, a lot of pollen, and they're storing in the cells where the queen should be laying eggs. That could trigger swarming. And then even genetics. So some stocks are just more prone to swarming than other ones. And so you may be buying a certain genetic stock or a certain queen, and based on their genetics, they may be more or less likely to swarm. And so bees can swarm for really a lot of different reasons. I guess one thing I didn't mention is diseases. And so if there's a lot of diseases or pathogens within a colony, they may be more likely to abscond and swarm. Now, swarming is really cool because half the workforce, about 50 to 60% of the workforce, will fly out with the old queen. So the colony will raise a new queen. Once that queen emerges, half the workforce and the old queen will fly out and find a new home. And so they work collectively together as they separate to find a new home and start building a brand new colony. So in a situation where your colonies begin to swarm, they'll raise a queen cell, and then half that workforce will try to find a new home. So what happens is that workforce will start flying above the colony, and it'll kind of look like, I guess, a little bit robbing to the untrained eye. It'll look like maybe a DCA or a mating area, but these workers will be kind of flying above the colony in a really frenetic motion. And they won't be there very long, but what they're doing is trying to orient the rest of the bees and really direct the bees to their new home. And so one reason all the bees are flying above the colony is really to direct and lead all the bees, like the 5 to 10,000 bees, to their new home. So after that, they might not be there for very long, and they'll try to find maybe a tree branch or a fence post or a structure to temporarily land on. And so a lot of times you might see a swarm near the colony. So, on a bush or a tree branch near the colony. And what these swarms do is form, I guess I like to call it temporary home, where these swarms are kind of clustering on a tree branch. Now, this is a temporary home. This isn't a permanent home because, as we talked about last week, the colony needs protection from the weather, they need protection from the sun, protection from the heat, and protection from any predators. And so they can't live on this branch for very long because if they do so, they risk a lot of different issues that can cause death to the colony. And so they sit on this tree branch or a fence post for maybe somewhere between 2 weeks or 2 days to maybe a week or so. But their goal is to find a more permanent home that has everything that they need to grow and survive. So on this temporary home, you'll have certain scout bees on the back, and they're sort of like the foragers, but instead of finding resources, these scout bees, their goal is to find a new home. So these 10 or so scout bees will fly out and find new potential homes. They'll come back and then they'll do a waggle dance on the back of this. And that tells all the workers about all the different options they have and a lot of different options about where their new home could be. Now they're doing that wiggle dance to tell the workers where that new home is. And the other workers will follow that dance and try to decide which home they want to go to. Now here's the catch. The catch is they all have to reach a consensus about what that new home is. And so if there's 10 bees doing a dance and 10 bees found 10 different homes, they all have to agree on one site. So if 70% of the bees are dancing with one bee site and 30% of bees are dancing on one bee site, then they haven't reached a consensus and they won't go to that new home. But over time, the bees are lobbying each other and trying to convince them to go to their new home. And so over time, that'll be 70%, 30%, 80%, 20%, 90%, 10%, and then 100% of workers will eventually dance with that one bee. Now, once the bees reach that consensus and reach an agreement of that new home, and this is really how democracy should work, where everybody agrees on it. And so bees do a really great job of having this democracy in place. But once they reach an agreement, they fly to their new home and start building the wax. The queen starts laying eggs, and they start building a brand new colony. And so these permanent homes could be in a lot of different situations. So I talked about this last week, but they can form an open air colony. I think this is a situation where democracy didn't work for the colony, so they start forming a colony right there. So maybe they didn't reach a consensus, so they start forming this open air colonies right on a tree branch. Now, like I mentioned last week, this is really undesirable for a colony because they're exposed to the environment, they're exposed to predators. Bears can easily access that and steal the honey. So this isn't a great situation for a honeybee colony. Tree cavities are a really great home for bees. They live inside trees. They can be pretty large. They have a lot of space to grow and move. They have, small openings. If they're larger, they can make them smaller. And so trees are a really great situation for bees, and it's highly protected. And so that's one situation, especially here in East Texas and certain parts of Texas, that bees will form these new homes in these tree cavities. And then finally, structures. So bees can form homes in, walls. They can form homes in, sheds or these other situations. And so these are really the three different swarm sites. But in order to go to these two homes or these three homes, they have to reach a consensus. And so we will talk about swarm management a lot more. So, especially when your colonies begin or when we start entering swarm season, we'll talk about different ways to, manage or eliminate swarming. But the best way is really to add space. So if you're 70% or 7 out of 10 frames are full of bees, that's a good time to add space because if you have more space in your colonies, your queen have more space to lay eggs. Your colony has more space to put in sugar or pollen, and that really eliminates or mitigates swarming. So really the best way is to add a super. But as we get into the swarm management section, we will talk more about different strategies to make sure your colony doesn't swarm. Because if it does swarm, you're taking basically away half the workforce, and that can make your colony weaker and more exposed to a lot of different things. And then, queen cells. So this is a sign that your colony is about to swarm. So if you see a lot of queen cells on the bottom of the frame, and they're not open like this, that's a sign that your colony is about to swarm, and you need to take immediate action before they swarm. If they're open like this, that's a sign that your colony has already swarmed or is about to swarm. So it's almost too late at this point. So maybe it's time to take take like a four-wheeler, drive around and try to find the swarm. Okay, so one of the coolest parts about honeybees is their social immunity. So honeybees actually have a very low immune system, especially compared to humans. So they don't have an adaptive immune system, so that makes them more prone and exposed to a lot of diseases. Also, because they're living in a confined space with 10 to 15,000 bees, they're more exposed to a lot of this disease spread. So you look at a lot of these different viruses, for humans, right? We go to daycares where there's a lot of, kids around. It's an easy way for these viruses to spread among a colony. That's the exact same way for honeybees because there's so many workers all in close confinement. It's really easy to spread these diseases. So one way that bees have combated this is through this social immunity. So that means is bees have these behaviors that allow them to combat these parasites, viruses, and diseases. So one of the social immunities is hygienic behavior. So hygienic behavior means bees have the ability to find dead or diseased brood and remove them from an entire colony. And so some bees have higher hygienic behavior and some bees have lower hygienic behavior. So it really depends on the strain. But this is something that beekeepers can actually select for. And so this is something that if you are buying queens, you can ask them if they're selecting for hygienic traits or if, they have in the past, because you definitely want your colonies to be more hygienic. And really hygienic behavior is one way for colonies to keep things clean. And the problem is if you have a lot of disease brood within your colony, like I said, because there's so many bees within a colony, those diseases and pathogens can rapidly and actively spread throughout a colony. And so bees really need this hygienic behavior to make sure things are clean and make sure things are hygienic and remove anything that can cause issues outside the colony. So kind of like a hospital office, right? So if you have some diseases that could be spread rapidly, they need to be removed or they need to be stored away so they don't cause issues for anybody else. Now, like I said, some colonies can be more or less hygienic, and so one way we test for hygienic behavior is by freezing 300 cells with liquid nitrogen, and that kills all the brood in this very specific area. We put it back into the colony and come back 24 hours later to see how many of these dead brood or diseased brood were removed. If more of those are removed after 24 days or 24 hours, that means that colony is more hygienic and has this very specific trait. So this colony is extremely hygienic. As you can see, this is all brood at some point, but all these dead and diseased brood were removed, whereas there's only a few cells that remained and weren't removed by the bees. And so this is 97% hygienic and something that you should be looking for as a beekeeper, or maybe not looking for, but something that you want as a beekeeper. This colony isn't very hygienic. So as you can see, there's a lot of open cells that were removed, but only 63% of the cells were removed. So there's still a lot of cells that were dead that the bees have removed. So this colony is really hygienic. This colony isn't hygienic. And if it's not hygienic, it's more likely to have diseases, pathogens, and parasites. Oh. So, one way we can test for hygienic behavior is this pin test. And so these have really small pins on them. And you push them into the brood, and it kills all the brood underneath those cells. And then you come back 24 hours later and see how many of those dead brood are removed. And so this is something that's really practical for most beekeepers. This thing is really cheap, easy to find, and easy to see if your colonies are hygienic or not hygienic. Now, you're probably buying queens, but it could be a good way to see if the queens you're buying are hygienic or not, and maybe find a queen producer or somebody that is actually breeding or has hygienic queens. The gold standard is this liquid nitrogen, but most beekeepers don't have access to liquid nitrogen, and it can be really expensive or time consuming. What we do is basically put these PVC pipes on a frame. We pour liquid nitrogen onto the frame, and that kills all the brood in that area. We put it back into the colony and then see if they're more or less hygienic. Now, this is the gold standard. This pin test isn't as accurate, but I think it can give you a baseline about how hygienic the colony actually is. So breeding, why should beekeepers actively select and breed for hygienic behavior? And the major reason is it's practically eliminated a lot of these diseases. And so we know that if you breed for hygienic behavior and colonies have hygienic behavior that's around 97, 95 to 100%, they are more likely not to get chalk brood because if they do get chalk brood, those are immediately removed. They're less likely and probably unlikely at all to get European foul brood, which is a bacterial infection, because bees are likely to detect that beforehand and remove it. But also, if they're removing it early on, it's like a spread throughout the entire colony. And then finally, American foul brood. So we talk about colony losses. This was a major reason why colonies died back in the 1900s to the early 2000s. But once we started breeding for hygienic behavior, this disease has practically been eliminated. I see very few cases of this, and one reason is because we're helping beekeepers select for more hygienic stocks. And so these bees are able to detect that and remove that from a colony. But this is all to say that, breeding and selecting for hygienic behavior is really valuable for removing and dealing with a lot of diseases. And so this hygienic behavior is a really unique and cool behavior that the bees have to combat a lot of these diseases and not expose the rest of the workers to these diseases. Another one is grooming. So, bees have this sort of grooming behavior where they're, going around, they're grooming the queen, they're grooming the other workers, and they're basically making sure that they're clean, making sure that they don't have any debris, they're making sure that they don't have diseases on them, and if they do, those workers could be and will be evicted from the colony. And so some workers have some sort of disease. Those workers are typically evicted from the colony because, the workers want to make sure it's as clean as possible. And if they have one worker that has a disease, then that could, really risk the rest of the workers. And so this grooming is really important for a lot of different reasons. And so I talked a little bit about grooming last week about how this is used to spread pheromones throughout the colony from the queen. It's used to make sure the queen is clean, but it's used for disease resistance and social immunity also. But one extreme form of grooming is what we like to call mite biting or mite hunting. And so typically grooming in a colony, and one thing you'll see is the bees will go through the colony and they'll clean each other off and groom each other off. But one extreme form of grooming is mite biting. And so when I was a PhD at Purdue University, we bred for this trait called mite biting. And this was ability of the bees to find mites, chew their legs off, and kill them. And so it's a really valuable trait and one thing that we use to breed for mite resistance. And this is what it looks like. So we collected mites on the bottom board. And as you can see from these lines, you can see some of their legs are chewed off. Even this kind of hard exoskeleton was chewed on. And it was just one way for once the mites emerge from the cells for the bees to bite them and remove them from the entire colony. And this is what it sort of looks like. So this is a worker that's bred for mite biting. They have a mite. It's kind of hard to see in this grainy photo. I'm hoping to get a better one. We can see that there's a mite in its mandibles, and they're chewing its legs off and chewing on the mite. Eventually, drop to the bottom board, and that kills the mite. And so this is a really valuable trait and one thing that we are using to breed for mite resistance and mite tolerance. And so one issue with this trait is it's really hard to test for. So what's different from the hygienic traits is we can freeze or kill certain brood cells, and that's a really quick and easy trait to select for. Whereas mite biting, we put a sticky board on the bottom of the colony, and so it has like oil on this. And so as mites fall, we can collect those mites and see what percentage of mites had their legs chewed off and what percentage of mites didn't have their legs chewed off. And so if we see more mites with their legs chewed off, these colonies have more of this grooming or mite biting behavior. But it's really time consuming and something that most beekeepers, don't have the time to do. If you're interested in trying and put a sticky board on to see, it's kind of cool to see if you do want to compare different stocks, but what I will say it is a bit more time consuming. One of the most valuable traits, most social hygienic traits is this real sense of hygienic behavior. And so as we talk about hygienic, there's different forms of hygienic behavior. So with the freeze test and the pin test, that is more hygienic behavior for sensing dead or diseased brood. And so that freeze test is a really great test for removing any diseased brood or bees within the colony. Now, real sense of hygienic behavior is really valuable for detecting real mites within the cells. And so what happens if mites are inside these cells and they're feeding on the brood produce a very specific pheromone. The workers know that pheromone. So they find that pheromone, they uncap these cells, and that disrupts real mite reproduction. And then they eventually remove the pupa, and then those mites are dead and unable to reproduce. And so this has been the most valuable trait that we've had to breed for viral resistance. Now, while grooming is a really great trait, and mite biting is a really great trait, it's hard to select for and something that's not easy to apply to a lot of different beekeepers. But VSH, even though it is a bit more time consuming, is one thing we can use to breed and select for, mite resistance. And so this is what we typically do is we encap 100 cells along the brood, we remove them, and we measure what percentage of mites inside the cells are reproductive or non-reproductive. And so it does take a microscope. It is a bit more time consuming, but we do know if we breed and select for this trait that bees will be resistant to Varroa if you are running a good breeding program, and they will be able to combat this Varroa. But a lot of times, if you are buying VSH stock, you'll see a lot of these uncapped pupa because a lot of bees are uncapping and recapping the cells because they're detecting whether there's mites inside the cell and then recapping them once again. And so if your colonies are extremely hygienic and extremely real sensitive hygienic, you'll see a lot of these uncapped cells within your colony. There was a recent researcher that wanted to find an easier way to select for this trait. So she's in University of North Carolina, Greensboro, and she found this pheromone and she's able to apply this pheromone on this space. And so that's one way that we are able to select for real sensitive hygienic behavior. We're still learning more about it. It's not as accurate as this test, but maybe it's a better step in the right direction. One thing that bees also use is social fever. And so just like when, we have a temperature, our body temperature is raised to combat these diseases and viruses. But instead of individual bees' temperature rising, the entire colony rises. And so what bees do is they try to maintain a temperature at 95 degrees within a colony. But there's an outbreak of a fungal disease, a bacterial disease, or a viral disease, the whole colony will increase their temperature in a way of combating these diseases. And one way they increase their temperature is by contracting their muscles in their thorax, moving them really fast, and producing heat within the colony. And this is how they produce heat to make it through the winter, but they also use it for this behavior called social fever. And so we're still learning more about this, but it's really cool if you do have like a brood monitoring device to measure temperature in the colony, and your colony does have diseases, you will see a spike in some of the temperature, and that's because the bees are using these methods to combat these diseases and pathogens. One way, another way is absconding. So if there's a lot of diseases in the colony and there's sort of a no return for the colony will swarm and abscond. And so that means that they won't leave the bees behind. The entire colony will fly out and find a new home. And so in these cases, and this isn't always the case because I think a lot of beekeepers assume it's absconding, but in some cases we have a lot of diseases. You may see honey or pollen stored in your colony, and that's because they had a lot of varroa and diseases, so they had to go out and find a new home. And this is more common in Africanized honeybees, and so those are more likely to abscond. But I will say that if you do see resources without honeybees, that doesn't necessarily mean meant they're absconded. Maybe they died and left the colony. But in some situations, the colony will abscond and leave. And then the final way, or one of the ways, is polyandry. So, like I mentioned, the queens will mate with somewhere between 15 to 20 males. Um, and one reason they do this is because it increases genetic diversity within a colony. So if they mate with more males, there's more diversity within the colony, and they're able to combat a lot of these diseases, pathogens, and pests. But one thing that happens with a colony if there's more disease within the colony or there's pesticides within the colony, the queen will actually mate more to increase genetic diversity within the colony to combat parasites, diseases, and even pesticides. And so we do know if, young queens before they mate are exposed to a disease or a virus or a pesticide, queens will increase their mating frequency from 20 males to upwards to 40 to 50 males. And this is some really cool research done at Texas A&M at Dr. Juliana Rangel's lab with Dr. Elizabeth Walsh at the USCA B Lab in Baton Rouge. She did some fantastic studies on this. So if you want more on that paper, I'm happy to send it to you. But, this is one way they can combat diseases. But also, if a colony is diseased, then the future queens that they produce will also, mate more often. So polyandry and extreme polyandry is one way that bees combat a lot of these diseases. And then finally, propolis. So I talked a little about propolis last week, but propolis is resin collected from trees. This has a lot of really beneficial substances to it. So it can be antibiotic, it can be antimicrobial, and one way that bees can really protect themselves from diseases and pathogens. So it's typically used as a glue to cover up any open spots in their colony, but it can be spread throughout the colony to really coat the colony and protect it from diseases from getting in. And so, typically Langstroth colonies when we buy aren't conducive for propolis production, but there's some really cool studies showing that if you go to the inside of a hive box and you ruffle it up a little bit, I think you actually buy some hive boxes that are ruffled up, then the bees are more willing to build this propolis and line the entire colony with this propolis. And it's just one way for bees to fight off an infection without the physical bees and social immunity to do so. Now, a lot of solitary bees actually use resin to coat their small cavities. So if you do find like a solitary bee nest and you see their nest, they may line it with propolis. And so because solitary bees don't have this social environment and don't have this social immunity, they use this resin as a really important way to combat diseases and infections. Another way is larval diets and eggs. And so one thing that bees do is they add microbes, they add nutrients and antioxidants and antibiotics to what the food is to the larva. And so if there's an infection in the colony, they'll increase these microbes, antibiotics, and other things into the brood so they can better combat these diseases later on. And so this larval diet is extremely important and a really important way for the larva and future workers to combat a lot of these diseases. And as I'll talk about the immune system, honeybees don't have an adaptive immune system like humans, right? We have antibiotic bodies and we can learn about new diseases. But really, bees don't have that. So one way they do that is by transferring these materials to workers so they can combat these diseases in the future. So instead of antibiotics, they really prime these larvae for a lot of different things. But queens can actually boost immunity of their offspring. So if a queen is exposed to a disease or a virus, they will put certain nutrients into the eggs so those offspring are primed and ready for the disease in the future. And so queens are really important and they change their egg quality so the young larvae and the young eggs are exposed to infection, but also primed when they become adults. And one behavior that I don't know much about and researchers at the community don't know about is washboarding. So typically what you'll see if it's really hot, there's a lot of colony bees in the colony, you'll see a lot of bees sitting in the front and then we kind of moving their legs and arms back and forth in kind of an up and down movement. And this is called washboarding. We don't know why they do it. We don't know if it's beneficial. It could be something to do with social immunity, so cleaning the front of the colony. We just don't know. But it's a behavior that we've known about for a very long time, but we don't quite know why bees do it. It may be to clean the front of the colony. It may be to put some kind of pheromone signal. We just don't really know. But I put it on here just so you guys are aware of a really unique behavior that us as a research community are still trying to figure out what it is. Okay, cool. So I think we'll take a 10-minute break. We'll come back at 11 o'clock. So that gives you opportunity to get some water, something to drink, and use the restroom. So we'll come back at